Hey guys, in this video we're going to be looking about some vocabulary that you need to be familiar with in order to hack your gene. The first word that you need to know is what a gene is, and everyone should be familiar with this. It is just a distinct sequence of nucleotides in a piece of DNA that is synthesized to form a protein. We'll be talking a lot about what a genome is, and a genome is simply a complete set of genetic material in an organism. In this sense, you will be told that you are hacking a genome, or you are annotating a genome that has multiple genes in it. And that is the goal, so be paying attention to those words. Also, nucleotides. So everyone's heard of a nucleotide when they hear ATGC. We all know that AT are paired together and GC are paired together in a DNA sequence. These are the chemical molecules that make up the rungs in the middle of the DNA ladder. As you can see over here, these are the rungs in the middle, and then you've got your two side pieces. So your AT and your GC are going right here in the middle. Each backbone, which are the side pieces, are lined with many nucleotides that are linked to other nucleotides on the opposite backbone. A single backbone and its set of nucleotides is called a single DNA strand. So this whole picture right here is one DNA strand. Because the DNA has two strands, it is called double-stranded. Okay, so like here's one side of the strand, here's another side of the strand, and then these two put together means that it's double-stranded, and this gives it that alpha helix shape that we call it, this twirling kind of look to it. Okay, so what are base pairs? You'll see this abbreviated as BP many of the time. It just stands for base pairs. All of the nucleotides are numbered from start to finish in the genome. This term is used when you are talking about the number of nucleotides, length of a gene, length of a gap or overlap between genes, or the location on the start genome. We'll use this term a lot, so be sure you're familiar with that. Okay, a codon. So a codon is 3 amino acids that are 3 nucleotides in each codon are read by the ribosomes, which are protein builders, to produce an amino acid, which is the building block of proteins. So, for example, you'll have TCG, and that produces the amino acid serine. So any like 3-letter, 3 sequence there, it'll make an amino acid, and these can be all different kinds of amino acids. Like this one, for example, GAG makes, I think that's glutamine, and then CCC is this pro one, and then every one of these makes a different amino acid. Okay, codons and starts and stops. So now that we know what a codon is, it's that 3-letter sequence. So a start codon means that this is the start of the gene. This is where it begins. So these start codons are going to be ATG, GTG, or TTG. And these are the only ones you'll ever see being a stop or a start. So pay attention to those 3. The stop codon is going to be AGA, AGG, and ATG. And ATG is both a start and a stop. Look at that. Pay attention to that. The problem with the start codons is that they can also code for an amino acid methionine, which is just a normal amino acid. So we need to determine which one's start and then which one is methionine. So make sure to pay attention to that. So what exactly is a protein? These are intermental units produced by genes that are perform certain jobs in our own cells, as well as the phages. When we assign functions to a gene, we are assigning a task to the protein that a gene produces. Okay, what exactly is a phage? And I know we talked about this in the beginning of the video for the intro video, but a phage is just a virus that infects bacteria. It's like the flu virus that infects our cells. A phage, or bacteriophage, is a virus that infects bacterial cells. Phages are found whenever bacteria exist because they need bacteria to live. It's kind of like a host type thing. They cannot live without bacteria. They need them to survive. You can find these anywhere in your backyard, your skin, your sink. I mean, they're all over the place. That's why, like, there's so many thousands of these in the world, and we're still finding more today. Phages have been used to fight off bacterial infections when antibodies don't work effectively. So not only are you, like, learning about genomes and all these things, but, like, this is real-world application. Okay, this video, I encourage y'all to go back and watch this video. The PowerPoint is available to you. I'm not going to play it right now. This really describes what a phage is, all the parts, etc. So if you want to know about that, I suggest you go back and watch this video. It's really interesting. Okay, so what exactly is coding potential? It's just the test that determines if a genome sequence contains coding DNA. Super simple definition. Okay, so what is a frame? It's a way of dividing a sequence of nucleotides in a nucleic acid molecule into a set of consecutive non-overlapping triplets, or codons. And then an ORF is strands for open reading frame. This is the part of the reading frame that has the ability to be translated. It is a continuous stretch of codons that begins with a start codon and ends with a stop codon, which we've talked about stop and start codons. Okay, a ribosome is just organelles found in both prokaryotic and eukaryotic organisms that bind to the mRNA to tRNA in order to create new proteins and polypeptides. So these mRNA and tRNA you should know from your standard biology class. So if those terms don't make sense to you, look those up, but that's in relation to a ribosome. Okay, Rbs and Scores, ribosome binding site. This is where the ribosome binds, hence the name. It's a sequence of base pairs at a specific location which call for a ribosome to link onto that location during the translation of DNA. Then the ribosome can go about its purpose. Scores. They're scored against the average scores to other potential RBSs, which gives us a deviation score, moment to or away from the average, at whatever is the best z-score is most likely to be a good start, but not the best determinant. So these z-scores, you're going to talk about that when you're annotating genes, and if you have more questions, you can easily ask your OBU partner about these, but it is a common term that you need to know. So why they matter. A score gives us a point in an argument of whether the start is good to use or if a gene is legit. So you don't want to, like, annotate anything that's not legit. So that is all for our terms. Like I said, go back to this PowerPoint that's available to you. Review these. There's also a Quizlet out there that's going to help you memorize these, and there's going to be a worksheet as well to test your knowledge about these. So these are just going to be used a lot when annotating, and I hope you found this video helpful.